When, in 1859, Charles Darwin published his famous book on the origin of species, this introduced the world to his theory of evolution. This stimulated many new studies on the origin of life and its diversification into all the millions of forms seen around us today. However, as soon as we mention the word evolution in relation to life on Earth, many people who believe in God feel threatened. This reaction is in large measure a natural response to the way in which the theory of evolution has been presented to us by some scientists. The first question to ask is, what is the origin of the first primitive life on planet Earth? How did this arise? In spite of very vigorous attempts to create life in laboratories, we have failed thus far, so it is still an open question. However, we can be sure of one thing, that even the simplest objects we call alive are nevertheless very complex. When mankind finally succeed in solving this question, it will be the result of the concentrated effort and mind power of many competent scientists over many years of research. We should keep this in mind and remain humble. We ask ourselves whether pure chance is the likely solution, since intense mind power and very sophisticated equipment has thus far failed. Our next question is, how did the first life change into the thousands of much more complex forms in existence today? We now know that our Earth contains about 3,200 species of mammals, 8,000 birds, at least 5 million types of insects, numerous fishes, amphibians, reptiles, and a vast array of plants. Darwin proposed that all of the great diversity of living things originated from one or maybe a few simple forms by a very slow process of change, one tiny step at a time. Darwin still gave some credit to God, as is shown from this quotation from the end of his famous book. I quote, "There is grandeur in this view of life with its several powers, having been originally breathed by the Creator into a few forms or into one, and that, whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. It is a sad commentary on the bias of some people that a later edition of Darwin's book, published after his death, changed this last paragraph of the book so as to leave out the words, 'by the Creator.' The present scientific understanding of the mechanisms thought to achieve this evolution is a combination of three main factors. The first factor is the observation that the genetic code of individual animals does differ. For instance, I look different from you because my genetic blueprint is a little different from yours. These differences are caused by each of us being the result of combining genes from both of our parents, and because the sexual cells used for reproduction also differ a bit in each egg and each spermatozoon from any one parent. As a result, there are many possible combinations of genetic material within any one species of animal and even between the offspring from one parent combination. That is why each child in a family normally looks different to the others. Secondly, in addition to the variations in genetics caused by different combinations of genetic material inherited from our parents, we do sometimes find apparent spontaneous changes occurring in the genes. We refer to them as mutations, and these can involve very small changes, such as a color change in these parrots. Small changes can be passed on to some of the offspring without causing any harm. Larger mutations, like this frog with 8 legs, usually die at an early stage in embryo growth, but occasionally such animals can survive. However, their chances of survival are small, and the chances of them breeding and passing on such mutations are even smaller. The third major tool of evolution is thought to be the effect our environment has on determining which of the natural genetic variants is most likely to survive. For instance, any natural variant living in a cold climate that is more tolerant than average to extreme cold is more likely to survive and produce its own offspring than is another individual that is less tolerant to cold. Evolution is not normally considered to depend on the suitability or unsuitability of one or two individuals in a population. It is considered by many biologists more probable that the environment acts upon whole populations of a species. Thus, in the example of penguins, over a number of generations, a significantly larger percentage of cold-adapted individuals will survive than the rest of the population. In this way, the whole population of animals in that one species can slowly become more cold resistant. It is not the cold that changes the genetics, but the cold acts to select which naturally occurring genetic variations survive. Parent stock that have a genetic makeup enabling them to survive cold better will tend to produce offspring with similar genetics. This is because, within certain bounds, these genetic traits are inherited. Thus, in time, the whole population will become more adapted to cold conditions. There are known examples of this sort of evolution. For instance, the peppered moth in England changed within 50 years from a pale colour, shown in the picture, to almost black, shown at the bottom left of the picture. This change in colour coincided with the start of the Industrial Revolution, when vast quantities of soot were deposited on buildings and vegetation. The explanation is that within the population of peppered moths, there were a very few individuals that had slightly different genetic makeup to the rest. This caused their wing scales to be black instead of a pale colour, and these few individuals had a higher chance of survival than their paler neighbours. Because of this advantage, the environment very rapidly selected for black wings. Black-winged moths survive better and produce more offspring than the pale-winged variety. Another way of studying this process of natural selection leading to new forms is to look at certain animals that are spread right around the world. A famous example is two gulls that coexist in England but normally do not interbreed. These are the black-backed gull and the herring gull. However, these two gulls are connected by a more or less continuous series of interbreeding populations distributed in a ring around the North Pole. If you move west from Britain, you see the change from herring gull to black-backed gull. In North America, they look more like herring gulls. However, they continue to vary as you make your way around the North Pole until, by the time you reach Siberia, they are starting to look more like black-backed gulls. When the extremes meet, as they do in Britain and Northern Europe, they do not interbreed. In Britain, they are now separate species. Other famous examples are the finches found in various islands in the Galapagos group. Isolated populations of one original species developed different characteristics on neighbouring islands, mainly with regard to bill size. This enabled isolated groups to exploit different food niches. The isolation on separate islands lasted long enough for each population to develop divergence in behaviour patterns, in addition to the differences in anatomy. As a result, when some of these isolated groups later reinvaded neighbouring islands, possibly blown there by hurricanes, some of them no longer interbreed. Their differences in anatomy and courtship behaviour effectively kept them as separate species. This inability to interbreed is one commonly held definition of a species. However, although they diverged in anatomy and behaviour, all these finches still shared the same basic genetics and still had the same number of genes. The environment had simply selected natural differences inherent within the genes. Since the environment and food availability varied from island to island, the genetic variants selected on one island were different from those selected on another. Lake Victoria in East Africa contains about 200 types of cichlid fishes, yet the lake system is less than 1 million years old. In 1990, Mayer and Associates published results of a genetic study. It showed that out of 14 of the fish types studied, all contained less genetic variation than existed within the human species. In other words, all these fish came from one stock and had diversified into different species by very slight changes within the same basic genes. In spite of such slight genetic differences, these 200 species some of which are shown here, are nevertheless distinct in their breeding behavior and so can maintain their different identities in spite of all living in the same lake. Inability of two closely related animals to interbreed is one recognized definition of a different species. Most people will admit that there is strong evidence for the formation of new races and even new species from similar existing ones. Plant breeders have many examples. However, we must ask, how does this relate to the much larger changes between simple life forms and more advanced forms? An assumption has been made by most evolution biologists that because we can see evolutionary processes operating between closely related species, this same process must explain the larger changes. Many textbooks on zoology and evolution outline the following approach to this question. First, because it is possible to see evolution at the species level, it is considered reasonable to expect that the same general processes extend to larger changes over greater time periods, including changes from simpler to more complex life forms. Second, the classification of life forms shows them to be in a hierarchical structure. In other words, we can place all life forms in families of similar creatures. Within families, we can place groups in one genus, and within a genus, we can place similar species together. In this way, we see so-called trees of life with increasing levels of similarity. It seems logical to speculate that all these creatures originally came from a common ancestor. Thirdly, the fact that nearly all life shares the same basic chemistry suggests a common origin. Furthermore, similar life forms generally have more chemistry in common than do forms that are less similar. Fourthly, higher life forms appear to show more primitive characteristics during early stages of embryo development. This is said to be like a speeded up repetition of more primitive life forms. In this diagram by Ernest Haeckel, he has falsified the evidence. He exaggerated similarities and minimized differences. I include his original fraudulent drawings because they are still reproduced in some biology textbooks. Looking from the top downwards, the developing human embryo is shown in the right column. This evidence, drawn up by Ernest Haeckel, has recently been substantially revised because of the fraud revelations. In fact, Haeckel was convicted of fraud by the University of Jena. Nevertheless, there are some similarities between developing embryos. Fifthly, some animals, such as mammals and mankind, have some organs that do not seem to have any obvious function in the body. For example, the appendix in man. These examples, often called vestigial organs, appear to be similar to structures that do have a major function in some other animals. Here we see a comparison in red of the similar structures in some other animals. These structural similarities are considered to be further evidence of interrelatedness. We now have the benefits of modern genetic studies showing how the DNA code of life varies very slightly between closely related species. For instance, modern man differs from the chimpanzee by less than 2% of the genes, and yet there are very marked and anatomical and intellectual differences. Both man and chimpanzees have the same amount of DNA in the genes of each of their body cells. It is the slight rearrangement of these so-called letters on the DNA that causes the observed differences between us. When we look at present-day life, we note close similarities between, for instance, domestic cats, tigers, leopards, and lions. However, these are very different from dogs or wolves. We do not see clear living intermediaries between such major groups. Evolution theory would say that this is to be expected, since the similarities would only be seen much earlier in the evolution of life. In some theoretical past, evolution theory would say that both the cat family and dog family should have had a common ancestor. They should have developed into two distinct families by a slow process of minute mutations and natural selection. A major question today is whether the fossil record supports such gradual change over time, or do we find much more sudden and larger jumps? The slow progressive evolutionary change over time theory encounters major problems. The typical picture shown by the fossils is sudden appearance of all the major animal and plant groups. On the diagram, the oldest rocks are at the bottom. Each major group of animals is shown as a vertical line, and we can see that each group appeared suddenly. The width of the columns represents the number of species. Although each group is represented in the fossils by many species, there is an almost total lack of intermediate fossils between each vertical group. For instance, the fossils show a sudden appearance of invertebrates such as crustaceans. These are creatures with an external skeleton and no bones. We are all familiar with crayfish and shrimps, and the species found alive today differ very little to those living millions of years back. Considerably later in geological time, we see the appearance of fishes. These are now creatures with internal skeletons made of bones. Fossils show a sudden appearance of many different types of fishes. Again, many of the fish species alive today are identical to those early forms. The same pattern is seen with the first appearance of amphibians. They also look much the same as many species are living today. After amphibians came reptiles and birds, and finally mammals. We also see in the fossil record the sudden appearance of other groups such as flowering plants, bats, birds, and in fact every new group of living things. All of these groups appeared suddenly. Furthermore, a growing number of biologists are finding evidence that breaks down some of the family trees drawn up by earlier scientists. In this issue of New Scientist Journal, we see the words "Darwin was wrong" and "cutting down the tree of life." This 24 January 2009 issue contains an article by Lawton called "Uprooting Darwin's Tree." In this article we read, "It is clear that the Darwinian tree is no longer an adequate description of how evolution in general works." The article asks, if you don't have a tree of life, what does it mean for evolutionary biology? At first, it's very scary, but in the past couple of years, people have begun to free their minds. However, the article stresses that downgrading the tree of life doesn't mean that the theory of evolution is wrong, just that evolution is not as tidy as we would like to believe. Another quotation from this article is from a researcher called Duprier, who says, "It's part of a revolutionary change in biology. Our standard model of evolution is under enormous pressure." Another scientist called Rose is quoted as saying, "The tree of life is being politely buried. We all know that. What's less accepted is that our whole fundamental view of biology needs to change, and facing up to this complexity will be as scary as the conceptual upheavals physicists had to take on board in the early 20th century." This article does not dispute that evolution takes place, but it does show that current ideas on how this takes place are now in the process of serious re-examination. The need for a new look has partly been brought about by careful examination of the fossil record and partly by genetic studies. This article in New Scientist Journal also refers to one genetic study that found one chunk of DNA in some animals that are very distantly related, but not in others where it might be expected. A peculiar chunk of DNA was found in the genomes of 8 animals: the mouse, rat, bush baby, little brown bat, tenrec, opossum, anole lizard, and African clawed frog, but not in 25 others, including humans, elephants, chickens, and fish. We would have expected such chunks of DNA only to be found in closely related animals or those on the same family tree.